The webinar is live now. Thank you so much, Chetan.
Thank you, sir.
Yeah, so we'll just wait another, it's still not 10 a.m. yet. We'll wait another 4 to 5 minutes and then we will begin the webinar. So we have 150 people already here. And the screen is shared. I would just like to ask all the attendees to put their questions in the Q&A box. Uh, as and when they think of the questions throughout the talk. And for those of you listening on YouTube, please put your questions in the comment. We will be monitoring them, and I will relay them to Professor Apurva Sarin in the Zoom call. With that, I will ask Chetan, the host, to stop the screen sharing, and I will hand it over to Professor Apurva Sarin to formally begin today's session.
Thanks so much, Chandrakant, Chetan, both of you. Welcome to everyone who's joining us on either Zoom or on the YouTube link. A very warm welcome, and we really are very heartened by your interest and willingness, I should say, to participate in these very, very difficult times. None of us are untouched, we know, with the pandemic, but I think the possibility of engagement and interaction, which is so important. This is, of course, a poor substitute for our meeting in person, but I think these are points of interaction and engagement. And we are very pleased to be able to continue this series that began in April. As some of you who have, I hope, now joined us for all the talks will know, this is the third webinar in the series. And as I had said at the very outset in our first seminar, when I introduced the Institute and our campus to you, that we are part of a larger cluster, which includes two other research spaces. And today we are very, very pleased, and I'm personally very delighted to welcome my colleague, Vatsala Thirumalai, who's a neuroscientist, and she is at the National Center for Biological Sciences, which is a center of the Tata Institute of Fundamental Research in Bombay. Vatsala, you will hear about her work, of course, in this seminar series today. She is a highly respected and recognized scientist and researcher in her field. She's also been fortunately recognized for all her contributions that her group has made to our understanding in the area. I am particularly delighted that Vatsala is here because she is also one of the faculties who has been a co-instructor on the neuroscience course, which is probably the longest running module on this campus. And it has played a very important part in integrating people and students who have very limited or no background in neuroscience to become, I think, some of the smartest researchers in the area. So with that, I'm going to hand over to Vatsala, and a quick reminder that we're still looking forward to your blogs, to your artwork based on what you're hearing in these webinars. Also, please send us notes about what you've been doing during the pandemic and how you've been coping with the stresses and strains of these times. So with that, I hand over to Vatsala.
Thank you so much, Apurva. Thank you for those kind words. Thank you for inviting me to be part of this very, very exciting Science Setu seminar series. And thanks to Chandrakant, Amrita, Vaibhavi, and the instrumentation team for putting it all together and making possible that I can connect with all of you online, even though in person is a challenge in these times. So what I'm going to do is I'm going to first stop my video just to conserve bandwidth. And I'm going to share screen so that we can go straight to the presentation. Okay. Yeah. Can someone confirm to me that you're able to see this slide?
Yes.
Yeah.
I think, yeah. Go ahead.
Okay, great. So today I'm going to be talking about what we've been doing in the lab using fish as model organisms. Of course, the biggest question that comes to our mind this morning is, why do we have to get up on a Saturday morning at 10 o'clock and listen to how fish do complex computations when there is a pandemic raging around us? And as Apurva rightly pointed out, it has touched all of our lives, and it has brought in so much of heart-wrenching news from everywhere, even when we hear about the suffering of people that we don't know. So why is it important to focus back on science? And the simple answer to that question is that we are able to fight the pandemic because of science. Science rules our lives, right? Be it looking at diagnostics, using RT-PCR or rapid antigen tests, or developing vaccines for it, or even understanding what the virus does to our bodies. Fundamental discoveries are those that have made it possible for us to fight it at this level, right? One cannot even imagine what would have happened if we had been totally unprepared for this virus. And so when people were designing these things, for example, RT-PCR, we were not necessarily thinking about its application for screening people in large numbers during a pandemic. The test was designed because scientists were interested in finding out how much RNA was there in a cell, or what kind of RNA was there in a cell. They were looking at it from a fundamental science point of view. They were asking why questions and how questions. How is it that a virus enters the body? How is it that the cell responds to certain antigens? How is it that an immune response is mounted? Why is it that some people get more sick than others, right? Not many of those questions may not even have had to do with human health. Many of the discoveries, for example, CRISPR-Cas9 that we hear about so much these days, came about because people were interested in studying how bacteria fight viruses, not necessarily how humans fight viruses. But then the applications that come from such studies, where scientists are purely driven by curiosity and they are trying to find out what is happening in the world out there, they find fundamental discoveries that then fuels applications later. So with that preamble, I want to just drive home this point that discoveries in basic science drive future applications, and therefore it's much more important for us to focus back onto our benches, work that much harder to figure out all of the different processes that happen in the universe around us, try to understand it as best as we can so that we can be ready for any challenge. Okay, so what is it that my lab works on? Broadly, we work on understanding how animals move. And today I'm going to be talking about one particular aspect of our work, and that starts with the question, can our brains predict the future? I mean, we've heard of astrologers drawing up charts and saying, you know, Saturn is in retrograde or Jupiter is in fifth house or whatnot. But does our brain draw up a chart and predict the future? It's not predicting the future in the same sense as an astrologer, but it does predict the future, you may be surprised to know. But it predicts the future because it's important for survival. Why? So one of the fundamental functions of the nervous system is to make animals move, and to make them move efficiently and quickly. And to do that, it's not just enough to be able to see what's happening at the moment and then move in response to that, because if you did that, you will be too late. Imagine a cheetah hunting deer in the forest. The cheetah has to predict where the deer will be in a future delta t point of time. It cannot go after the deer where it is at this point of time, because if it did that, it will be constantly chasing the deer and never reach the spot where it will be at the future, right? The trajectories have to intersect. And that's why predictions are important. So there are many aspects of prediction that the brain does. For example, look at this picture of this person jumping from one place to another. To be able to do this feat, you have to predict what kind of forces you will generate with your limbs, and what is the effect of gravity, how fast is it going to pull you down, and then calculate your trajectory from this endpoint to this endpoint. If you miscalculate, your endpoint will be somewhere there, and that's counterproductive for survival. And this is just one example where, you know, the body is thinking without you knowing. It's an unconscious planning action that the brain does, and it is able to generate a movement plan And then that plan is executed so that the organism, in this case, the human, is able to jump from one place to another without hurting themselves. And this happens out in the natural world all the time, right? Animals navigate their environments, be it sharp rocks like this, or slippery slopes, or even water for swimming animals, water currents. All of these things are constraints that the physical world imposes on animals, and the animals have to learn the rules of this physical environment. You can imagine if tomorrow we colonize Mars, then we'll have to relearn the rules of Mars to be able to live there. The rules of gravity that apply in Earth do not apply on Mars, and the rules that we learned here will not be easily applicable or straightforward applicable in a different planet. So that's one kind of prediction. Another kind of prediction is also what's happening in the outside world. For example, if there is a repetitive stimulus, look at these two kids playing jump rope, right? And this little kid over here needs to learn how frequently is her sister throwing the rope or around her, and then time her jump at the time that the rope meets her leg. If she doesn't time it appropriately, then she is not able to jump on top of the rope. Of course, there is a learning phase involved. Initially, the little kid will not be doing as well. But with time, if the sister imposes the same sort of rhythm repetitively, then she's able to learn that rhythm and respond appropriately. Now, my talk is going to elaborate more on predicting the timing of stimuli so that appropriate motor response can be planned. And this is key because many times around us, we see visual stimuli or mechanical stimuli that occur in a repeated fashion with a more or less fixed periodicity. For example, let's take playing tennis, right? The ball keeps coming to your side of the court at a regular time interval, and you should be able to predict when it's going to hit your side of the court so that you can meet it and give a response. So what I'm going to tell you today with that preamble is that brains use current inputs, that is experience, to predict the future so that the response is quicker and better, so that organisms navigate the world around them in a better way, and it also helps them survive better. And this work was done by a brilliant graduate student in the lab, Sriram Narayanan. And as Apurva rightly pointed out, many of our students come to campus without any neuroscience training because there aren't that many neuroscience courses in the country. Sriram is one such student. He has a bachelor's in biochemistry, but he came to our integrated PhD program, and you will be astounded to see what he has achieved by himself working in my lab, acquiring all the skill sets that are needed to put this project together. Okay, so our model organism are zebrafish larvae, and we use them because they are transparent. We can look at their brains in intact organisms. They don't have a skull, so we can see what the neurons are doing in the live organism. You'll see example of that in the coming slides. And these fish live in shallow moving streams of water, but they want to stay in one place. They don't want to be washed away by the water current. And so they have this reflex behavior called optomotor behavior. What is it? Imagine yourself on a treadmill, right? You're looking around. If you're standing on the treadmill and the treadmill is moving, the world around you will recede from you, right? You're going back and the visual scene is going forward. Similarly, if the fish sense that the visual scene is going from the tail to the head forward direction, then they generate a swim response to compensate for that flow, and then they try to maintain their position with respect to the moving visual scene. And we can easily see this in the lab by giving the fish visual stimuli. So what we've done is in this box now, you see little things. These are zebrafish larvae, and we've put them in a tank of water, and underneath is a computer monitor, and we are going to show black and white bars moving from right to left like this. And now you will see in this frame, the fish are uniformly distributed across the entire tank. See what happens when the grating starts to move. The black and white bars are called gratings. And now it's going to come on any moment, right? So it's moving from one end of the tank to another, and the fish are sensing this as the water receding in one direction, the current flow in one direction. And you see they've all aggregated on this side of the tank more or less. So they sensed the flow in this direction, and therefore they've aggregated here, right? And this is called optomotor behavior. And we can also induce this in larvae that are head fixed. And that's important because, like I told you, we are going to look into the brain. And if the fish is freely swimming around, we can't look into the brain through a microscope. And therefore, what we've done is we've stuck the head of the fish in agarose, a jelly-like compound. The tail is free to move, right? And now we are going to show the same gratings, the black and white bars, and see what happens. And see, the bars are moving in a tail to head direction, and the fish is compensating by producing swims. These flicks of the tail from one side to another are swims. And this is the optomotor behavior that we are studying. And what we do next is that we make a custom microscope where we can put the fish head embedded, like I showed you in the previous slide, have a screen that shows these black and white gratings, and then we use laser light to image the activity in the brain. I'll tell you more about that in a bit. And what we also did was a clever trick, which is that, you know, you want to make sure that the fish thinks that its swimming is making a difference. If you just let the fish sit there with the gratings flowing all the time, the fish will give up after a little while. So what we do is something called closed loop feedback, where we image the movement of the tail. I'll just stop that video for now. We'll get to that. So we image the movement of the tail using cameras placed underneath. And then we have computer algorithms which can detect motion. And then we call based on threshold whether, you know, the tail moved and made a swim or not. And depending on the intensity of the swim, we then move the grating appropriately. It is to give a sense to the fish that when it's wiggling its tail, the stimulus is getting modified appropriately, right? So that the fish feels that its swim is stabilizing the grating. And that's what that looks like. See, when the grating moves, the fish swims, and then the grating moves back. And then again, another trial is done where the grating moves, and then the fish wiggles its tail, and then the grating is restabilized. And we do this in a very interesting sort of experimental trial structure. What we do is we place these optic flow stimuli in regular repetition. Remember the jump rope example I gave you? So what we do is we have optic flow on for 2 seconds, and then off for 5 seconds, then on for 2 seconds again. And like this, we repeat these trials about 200 times, okay? And so whenever the optic flow is on, the fish is making a swim, right? But once in a while, we play a trick on the fish. When the optic flow is supposed to come on at a certain time, we withhold it, and we call it a probe trial, and the grating stimulus remains stationary, right? Fish does not swim because the grating is not moving. It's just a stationary bar like this, right? And these we call probe trials. And we do this because we want to ask, okay, you're giving these repetitive optic flow stimuli to the fish. Is the fish learning the pattern of the visual stimulus in that it comes on every 5 seconds? Is it able to learn that pattern? And the quick answer to that question is yes. The fish are learning the pattern of the optic stimulus, which is kind of, sorry, 1 second. So the fish are learning this pattern because when the probe trial happens, then after the probe trial, the trial immediately after the probe trial, the fish are a little bit sluggish to generate a swim. So if you imagine yourself doing a task like this, where let's say you're asked to respond to a beep by clapping, and you're clapping along with every beep, and suddenly there is no beep, right? And you don't know, if there is no beep, then you don't know for the next trial, should I clap or should I not clap? You have a certain uncertainty that creeps into your mind, right? And that uncertainty shows up in the form of an increase in latency in the next trial. So latency means the time taken from the onset of the visual stimulus to when the fish is making a swim. Okay, so usually it's very quick, like you see here. So that's when the optic flow stimulus comes on, and that's when the fish swims, and it's very quick. But right after a probe trial, the latency becomes a bit longer, but significantly longer, right? And this shows you that the fish does have some knowledge about when the probe trial, when the optic flow should have come on, and upon not seeing that, it's getting a little bit jittery. Now, the interesting thing is we can lesion, or we can take away a certain part of the brain called the cerebellum, which some of you may know. It's at the base of your brain. It's important for producing coordinated locomotion. And it's thought to be the place where all such predictions are being deposited by sensory stimuli, right? So we lesioned the cerebellum and asked whether the fish feel the same jitter or the uncertainty after the probe trial, and it turns out that they don't, which tells you that the ability to predict and to modify the next motor response, the next swim, lies somewhere in the cerebellum. Okay, that's the lead that we got. So then what we did, we have, so focus on the bottom portion of this slide here. There are genetically engineered zebrafish where we can label specific neuronal populations, in this case, Purkinje neurons in the cerebellum, which are very important neurons in the cerebellum. So we can label them with a certain kind of sensor. In this case, it's a calcium sensor. And the sensor will glow brightly when the neuron becomes active. So you can assay the activity of many hundreds of neurons at the same time. And there are other groups out there in the world who can image the whole brain of the zebrafish under situations like this. We are also getting ready to do such experiments. But for this study here, we are focusing on the cerebellum, and we are sampling these Purkinje neurons with calcium sensor genetically engineered into them. And we are asking, what happens when a probe trial happens? When you give a probe trial, how do these neurons respond? What happens when a flow comes on, and so on and so forth, right? So this is what an experiment looks like. You have the Purkinje neurons down here. We are imaging through the 2-photon microscope. Through the other camera, we are monitoring the tail movement, right? And simultaneously, we are also giving the grating stimulus, the black and white bars. And these traces that you see on the right-hand side, each row is one Purkinje neuron. It just shows, you know, a sub group of them here, but easily in every experiment we can get anywhere from 40 to 80 neurons that we can image in one plane, right? And you can see that here, you know, the neurons glowing brightly and dimly as the fish is doing some task or another. And the traces here are just a quantification of the fluorescence brightness. So whenever you see a sharp peak like that, that means that at that time point, that Purkinje neuron made a very nice bright calcium signal, right? So now we have these data. What is the data? We have three kinds of data. One is the optic flow stimulus that we are giving with the timing of the probe. So anytime you see a gap, big gap like that, that is a probe trial, right? So this is what we present to the fish. This is the visual stimulus, the grating, when it's on and when it's off and when it's probe trial. This is one stream of data. The second is the swim velocity data. Like I told you, we were imaging the tail, so we can quantify when the tail was making a swim. So each uptick here, each of these is a swim bout, and the height of the peak corresponds to the speed with which this fish was swimming, the swim velocity, right? And the bottom trace is just one example of one Purkinje neuron and its calcium activity. So when did this neuron become active? What are the times at which this neuron showed very high increase in electrical activity as reported by the calcium sensor? Okay, so then we can do some computations, data analysis, very simple. So what you do is you chop it up, this trace, and you align with respect to either swim starts or optic flow starts. And then you ask, what did the cell do at that time, right? These are called event triggered averages. The event being either the flow on or the swim, okay? Or you can look at the probe. For example, the trials where let's go back here. So this would be a probe start, for example, the time at which the optic flow stimulus should have come on but did not come on, right? So we have these 3 or 4 different kinds of event triggered averages that we can make. And for every cell, we can make an average like that and ask, what is the average behavior of this cell? Did it like to respond when the optic flow turned on? Did it like to respond when the fish made a swim bout? Or did it like to respond when the probe trial happened? And the last category is what we are very, very interested in, right? So when we did this analysis, so first of all, very simple, this left-hand side graph, the color here, the warmer the color, it indicates higher level of calcium activity, okay? So 0 is when there is no calcium response, and 1 is when there is a lot of calcium coming into the cell, indicating that the neuron was electrically active at that time. And the red line indicates when the flow came on. That means the grating started to flow, right? And the rows here, each row is a cell, and we collected about 1,800 cells from different fish, right? These are data from about 27 fish, and it's all laid one on top of another just to see what the trend is like. And you see that there are, most of the cells are responding to flow on by an increase in activity. So you can see that they are blue right before the trial starts, and that they become yellow when the flow starts, right? And these sensors are a bit slow, so that's why there will be a lot of lag between when the flow comes on and when you see the signal. But what was more surprising to us, was that in the trials where the flow did not come on, that is the probe trial, and the red line again is the time when it was expected to come on based on previous experience. See, remember the fish was seeing these optic flow stimulus pattern, 2 seconds on, 5 seconds off pattern, and then it learned it, right? And so when when the flow is supposed to come on and it didn't during a probe trial, there are some neurons which report it. And you have this signal here. See this yellow signal here, right? And that yellow signal signifies that these neurons are saying, hey, come on, there was supposed to be a flow trial, a flow on, but it did not happen. And that's what the response looks like when you plot the amplitude of the fluorescence. DF over F just means change in fluorescence. It just became that much more brighter when the probe trial happened. And what are these cells then reporting? What are they reporting? They are saying that at this time point 0, I must have seen the flow turn on. I must have seen that the grating was starting to move, and then I must produce a swim response, but I'm not seeing a flow, right? It's reporting an error. And so this is called a prediction error, because the fish is making a prediction and it's reporting an error based on that prediction, saying, I predicted that there would have been flow here, but flow did not happen, and therefore this is an error, right? And why are these prediction error responses very interesting? First of all, they tell you evidence that the fish has learned something. Otherwise, there is no reason for having an electrical signal in the neurons at this time point, which is, it was tightly linked to the time of arrival of the probe trial, right? So this is clearly evidence that the fish has made a prediction based on past experience. But there are other cool features about this error response, which is that it is long lasting. You can just keep. So what we did was we gave them a bunch of probe trials in succession. Means the grating was kept stationary for a long time, about a minute or so. But this response did not decay. So these cells reported an error for multiple cycles, not just the first time that the probe trial happened, but they had a clock of the 5-second interval, and every 5 seconds they said, oh, the flow did not happen, the flow did not happen. And they did it for about a minute, right? So that's a long lasting memory. Even though there is some decay of the amplitude of the response, that's a long lasting memory of past experience, saying that I learned this pattern, but the pattern is not happening. The other cool feature about this is that even though it is long lasting, it is not set in stone, and that's beneficial, right? You don't want to be someone who learns one pattern only and then you don't want to forget it, even if you don't encounter it in your life again. You want to be able to forget old patterns that are irrelevant and be able to learn new patterns. And that's what these Purkinje neurons are doing. So here, what we did was we gave them 60 trials of the 5 seconds trial interval, and then suddenly we switched to 7 seconds intertrial interval. So this is a new pattern, and the prediction error signal also shifted from 5 seconds to 7 seconds. That means that these fish learn patterns. They're capable of learning patterns of sensory stimulation that they encounter, but they are also able to change those patterns, adapt those patterns when conditions change. And that's a key trait for successful survival in the world, right? So why, I told you that these prediction errors are very exciting. Why are they very exciting? They are very exciting because they underlie an animal's ability to learn patterns of sensory stimulation and respond effectively. How do we know that? So we analyzed the swim performance of larvae across different trials. So this is a slightly different analysis. What we did was this is example from one fish in 2 different trials. See, the neurons don't all respond all the time, right? They have some probabilistic response behavior. So we took trials, 2 trials. So here are 2 trials, one in the top and one in the bottom. And again, the rows are different cells, neurons, Purkinje neurons, imaged. And the red line is the onset of a probe trial, okay, the time at which flow should have come on but didn't. And there is in this trial, in this particular experiment, there are a large fraction of Purkinje neurons reporting a prediction error. And that's signified by this probability number 0.26. That means about 1/4 of the neurons responded for the prediction error. Okay, but then there are, in the same fish, there are other trials where there are not many prediction error responses. It's an animal, right? So it doesn't respond the same way from one trial to another. Every trial has a certain probability associated with it. So in this bottom trial here, you see that there are not many cells showing yellowish activity, right? And so the probability of seeing a prediction error response in this trial is 0.04. It's a low number. So then we asked, from trial to trial, if an organism or an animal has prediction errors of varying strengths. So the top would be a strong prediction error signal and the bottom would be a weak prediction error signal. What does that mean behaviorally, right? And one output that we can monitor is how quick the fish is able to respond at these different trials. So we look at the response latency. Once the flow comes on, what happens, how quickly is the fish able to generate a swim response. And because there is no swim for the probe trials, we looked at trials that are close enough to the probe trial, okay, to ask, does it mean that the fish has greater confidence of the pattern, probability of responding prediction errors? And that's what it turns out to be. So let me explain that in a little bit more detail. So here is a trial where the P probability was 0.24, a high prediction error probability, and the latency to respond after, so this purple color indicates when the flow came on, okay? And you see that the swims are generated very, very quickly after the flow is turned on, right? Similarly, here in another trial, the probability is 0.26 and the latency is also very, very short. Soon after the flow turned on, the fish was able to mount a swim response. Compare that to trials where the prediction error probability was low, 0.08 or 0.04, then the latency is also much longer, right? So that means the fish was slower to respond when it was showing lower prediction error probability. What does that mean? So what it means is that the fish has an underlying understanding of when flow is supposed to come on, and following that understanding, if the flow keeps coming on, then it's able to generate responses quickly. And if the flow doesn't come on, then it generates a very high prediction error response saying, hey, it didn't happen like I expected it. Contrast this to the situation where the fish has a weak model. It doesn't is not able to predict the flow pattern in that much with that much confidence. Then its response to the when the flow comes on also comes very slowly because it's not able to predict, right? So it's not able to plan. It's not able to plan for when the flow comes on. How, you know, I have to make a swim. So that's a weak model in the bottom row. It's not, it doesn't have an expectation of when the flow should come on, and therefore the swims are also sluggish, right? So that is the summary that, you know, when animals encounter repetitive stimuli in their environment, they learn that pattern, right? And they make an internal model, which is the expectation that I talked about. So they make a model saying, you know, this is how the stimulus keeps coming every 5 seconds or every 7 seconds. And when it comes on, this is the kind of swim that I have to make. And when they encounter stimuli, they compare that stimuli to the internal model and they ask, okay, does the input match this internal model that I have? If it matches, they say, okay, strengthen the model, keep this model works. Your prediction is correct. But when it doesn't match, like when we gave the fish the probe responses, the probe trials, then the fish's underlying confidence in this internal model is rattled. It says, hey, you know, it doesn't match this model that we have. So weaken that model, learn something new, right? Which is what happens when we gave it the 7-second stimulus. So with that, then I will stop my talk. You know, so fundamentally what I've told you is that even in lower vertebrates like fish, there is evidence of the brain predicting what's happening outside based on previous experience and using that information for better adaptive responses so that they can survive better in the wild. So that is my group. This is Sriram, whose work I talked about. Jagdish is our fish facility manager. helps take care of the zebrafish, and we get funding from all of these different sources, and of course support from all the facilities on campus, which enables us to do this kind of work. So thank you.
Thank you very much, Matsala, for that. I think you took us through a very complex set of work and simplified it enormously. I can tell you that I could really follow everything that you said, and I hope that this worked well for our students as well. So while we wait for the questions to come up, I'm just going to ask you, probably take the advantage of being the moderator. You brought up the example of the cheetah and the deer at the very outset, right? And so that is, of course, I mean, visually also, we know that this is a much larger, complex system. So the question here is, how does work in model organisms, such as zebrafish, enable our understanding of animals which look seemingly, where the brains might or might not be more complex?
Right. Wonderful question. So the reason for working with organisms like zebrafish is, one, of course, I talked about the transparency and our ability to peer into the brains while the animal is doing its thing, right? Non-invasively. We are not cutting open, making surgery or anything like that. So these kinds of experiments that I talked about today are not easily done in larger animals. That is one thing. But the second advantage is also that the zebrafish, being a vertebrate, many of the brain regions that you find in the fish are conserved from fish all the way to humans, cerebellum being a key example. So, what we find in the fish, if it's true in the cerebellum of zebrafish, it's most likely to be true in the cerebellum of humans, adding some more complexity and numbers to it. The third advantage is that, because it has fewer number of neurons, it's possible for us to do this sort of analysis where we are sampling. So I told you we sample about 60 to 80 neurons, Purkinje neurons in the cerebellum, and that's a significant proportion of all the Purkinje neurons in the cerebellum of zebrafish. There are only about 300 to 500 neurons. But if you take a mammal, we won't be able to sample a very large proportion of them. That means that, how do we know that what we are seeing is representative of what is happening in the other parts of the brain or in the other parts of the cerebellum, right? So that this gives us a picture of the whole much better than what we can do with larger organisms. So right. Those are some of the advantages. Yeah.
Certainly. Yep. Okay, we have a few questions coming up. I'll hold back on the others. So we have, again, just a note to us, people who are posting questions. We understand that sometimes you don't want to identify yourself, but if you would let us know your college, it would be wonderful, but let that not hold you back. Go ahead and ask the question. That is more important. So, Vatsala, the first question that we have here is, how is the prediction error identified? Someone has been listening closely.
Good, cool. So what we do is, I showed you this trace of the multiple flow trials and the probe trials, right? So we chop up the entire trace and we align all of the probe trials one below the other. And then we go to the calcium imaging trace, right? And then we see, okay, for all of these probe trials, how did this neuron respond? And then we calculate the event triggered average, which is the probe triggered average. And if that average passes certain statistical tests, like, the amplitude should be 2 standard deviations or 3 standard deviations away from the baseline noise, then we call it a probe response or a prediction error response. And we say that this neuron was responding because the flow was expected but did not occur, and therefore it's a prediction error response.
Okay, so we now have another one, which is a bit different and away from the experimental system. So it says, sometimes we get visions and dreams. Would that also be our brains trying to predict the future, or I suppose build on experience as we sleep?
Very interesting questions. Yeah, so I mean, we get dreams of all sorts, right? But In many cases, it is our brain trying to make sense of organizing our experiences during our waking periods. There are very interesting experiments in mice running through a narrow, what do you call, an alleyway or something. And they are recording from neurons within the hippocampus which is a region of the brain from the mouse. And when the mouse is sleeping, the same pattern of activity happens, but in crunched up time. So what in waking time, if the neurons fire in, let's say, neuron A fires, then neuron B fires, then neuron C fires in about a minute, when the mouse is sleeping, same pattern, neuron A, B, C, but happens in much shorter time window. It's as if the mouse is compressing its experiences and learning from it. Of course, we can't tell if the mouse is dreaming about going in those tracks, those alleyways that the experimenters built for it. But this is one example. But also, mental repetition, mental practice is something that has been shown to work for motor learning, be it tennis players or piano players, complex motor learning, even dance performers. Even if you can't do it physically, they do practice it mentally, right? Even cricket players, for example, how they throw and how they bat and all that. If they do mental practice, that actually makes their performance better.
We will have to get you back, Vatsala, to hear more about this. Anyway, I also just have a comment for my request to my co-moderators. Please don't remove the questions from the chat box, just leave them be, even if they are answered, because sometimes we go back to them. The next one is from Manish Bhatt. So I'll just read out the question. If a fish was exposed to the stimulus for a certain time period and then left alone for a few hours and then exposed again to the same stimulus after the interval, how long would it take for the fish to recall the previous pattern? Or let's read again. It's the same stimulus being shown twice, I suppose. That's the question, right? If the fish was
with a gap of an hour or a few hours.
Yeah, within a time interval.
Yeah, so we've sort of looked at it not at hours times, but 10-minute blocks. And it turns out that the fish can remember after about 10 minutes. It'll be interesting to look at it, over an hour or, from one day to another and so on and so forth. We haven't done that.
Okay, then we have a question from Swapna from St. Joseph's College. In the experiment, we observed that there is a lack of a lag period during the probe trial when the cerebellum was removed. There is a lack of the lag period.
She means the latency modulation. So yeah, okay.
The probe trial when the cerebellum was removed. Has this been associated with a particular region in the cerebellum, and is that identified?
Right. So that was not a lack of a lag period, that was the lack of modulation, meaning that the fish is not changing its latency based on the prior experience. It's not building in an uncertainty. So the uncertainty and prediction error comes into play because the cerebellum is there, and the cell type involved is the Purkinje neuron because it's the principal neuron type in the cerebellum. That's why we imaged these neurons. Of course, there could be other players. We do know that the inputs coming into the cerebellum carry information about prediction errors. So it's a complex circuitry. It's not just one cell type or just one part of the brain. Although we've focused on cerebellum, the cerebellum is talking to other regions in the brain, and the other regions are providing inputs as well. So it's the interplay between multiple centers. But what I wanted to show today was that these signals exist. And the place that we looked at was the cerebellum. If we look elsewhere, I'm sure we'll find it elsewhere as well.
So this is again connected from Anjali. It says it was mentioned that the cerebellum was associated with movement. What experiments were done to show the same? Could any other region be responsible?
Yeah, like I said, it's complex interplay between multiple regions. For example, we are showing visual stimulus, right? And the optic tectum is needed for looking at visual stimuli. And so if we did something to the optic tectum, this response would be abolished. And the optic tectum then transfers information to the cerebellum. And the cerebellum then talks to multiple centers within the brain, including the brainstem areas, which then talk to the spinal cord. So it's like a symphony that's being played out. What the cerebellum does is that it has these signals that show that the fish is making a prediction. Where is that prediction coming from? We don't know. It probably is coming from outside the cerebellum, but we don't know the regions that are involved there, and those would be targets of future experiments.
So again, related, Vatsala, I think to this the question is being asked here is that, is this phenomenon related to habituation, or is it the same?
So this is a reflex response. And so it doesn't really habituate down as long as we give it the sensory input back, which is why we did the closed loop design. So you show the grating, the fish responds, and then you move the grating according to the fish's swim response. If you keep doing that, the fish can keep responding for a very long period of time without habituating. But if we don't do the closed loop, if we leave it open loop, which is you show the grating, the fish swims, but nothing happens to the grating just keeps on flowing, the fish gets into a state of fatigue or giving up and saying, you know, my motor response, my swim response is not making any difference to the environment, so I'm just going to give up and not swim. It's not really habituation, but it's called learned helplessness or something like that. And glia are involved in that response. Other groups in the world are looking at those responses, but habituation does not happen for this particular kind of response.
Do you want to just explain habituation to the group?
Yes, habituation is when you have a sensory stimulus and the animal makes a response. Typically, these are noxious stimuli, and the animal does a protective response. For example, let's say you're poking the fish doesn't escape away. If you keep on doing it, the fish or any organism for that matter learns that this stimulus, which is happening at a low threshold, which is not harmful, then it learns that there's no need to respond at such a high level, and therefore the response amplitude decays over time, and that's called habituation. On the other hand, the opposite will be sensitization, where with experience the response amplitude goes up, and that's called sensitization. And there are multiple neural mechanisms involved in both of these patterns that we see.
So we've got lots of questions coming in, Vatsala. So first I should add that there's a note of appreciation, which comes from one of the teachers to say that it was really thanking you for demystifying or simplifying how one asks questions in a very complex area. But that was a comment. But we now also have a question from Glavin Rodriguez, and I will read this out. It says, I believe that fish can recognize the same stimuli even after a span of few days. So for instance, if we feed the fish at the same spot for several days, then the fish will automatically come to the spot at the same time on consecutive days. What is your view on this?
Yeah, absolutely. So this is another kind of learning called associative learning, where the fish learns. For example, it can be a place, or it can be a light. For green versus red light, you can put in different parts of your tank, and you can always associate the green light with food and red light with nothing. And when you shine the green light, then the fish will come to that spot for in expectation of food. So they are able to. Such learning is found in even simpler organisms. Even in invertebrates, Drosophila, for example, you can have them associate two different stimuli and say that okay, this is so when this comes on, then automatically with a certain delay another thing comes on, and that thing is either beneficial or noxious to me, and either I should go towards it or I should run away from it. These sorts of associative learning paradigms are present ubiquitously across the animal kingdom. Yeah.
And you can actually map them and try and understand.
And we can map, and in many cases we do understand what the neural basis of how this works. Not to the full extent, but we do understand, what neurons are involved, what is modified when the animal learns, and so on and so forth.
Correct. Okay, so again, a note from Alan Stricker. I think I've got the name right. A wonderful talk, thank you. Fish are thought to have low short-term memory, although recent studies seem to disprove this. How long do fish remember the stimulus in your model system?
So this particular experiment, as I said before, we've tested with a gap of about 10 minutes, not longer, because of the constraints imposed by our imaging system. We can't expose the fish to laser light for very long periods of time. It'll kill them. But I don't know if, there are paradigms where fish remember for, over days. That's something that needs to be looked into. What is the extent of memory? How long do they remember?
Okay, we have many more questions related to the element of your talk, but I will pick up one question from Swapna, which relates to your answer, Vatsala, to the visions of the dreams, right?
Mm-hmm.
The comment she makes here is that isn't tennis and dance, etc., related to muscle memory? She said this is the answer given for the dreams question. I think she's asking for a clarification here.
Mm.
So the concept of muscle memory.
What is muscle memory is basically, how elastic the muscles are by stretching them and so on. But complex movement patterns, muscle cannot by itself remember a pattern. The muscle can simply stretch or contract with electrical stimulation and stretch passively. It cannot really remember complex patterns that are involved in dance, such as moving your hands in left hand this way, right hand this way, and then coordinating with the leg movements. All of that comes because of the nervous system. And so the learning part of that comes from the nervous system, not so much from the muscle. Okay. Not at all from the muscle.
Okay, so now we have someone from the University of Calcutta, Arnab Ray. Okay, mm-hmm. Welcome. We are certainly reaching more than our original list. Question here is about, now I think goes back to your predator-prey example. Yeah. So mm-hmm. The question is that, is it the same error that is made by the prey? And here let me read it as it is written. Ma'am, is it the same error made by a gazelle that makes it a prey to the cheetah, or that mm-hmm. makes the cheetah miss its prey based mm-hmm. on the pattern of hunting and escaping that goes on, in the wild?
Absolutely, absolutely. So it's ultimately about who is the better predictor, right? For example, if you have cockroaches in your house and you happen to see it and it runs away, the startle response is never in a fixed direction. Every time it will choose a random direction to go away. And this is true of multiple escape responses across the animal kingdom, where the escape trajectory has some variability associated with it. And this is true of the deer also. Running away from the cheetah, it will make random zigzags, sudden turns, so that the cheetah is not able to predict what it's going to do next. But this is why young animals, young hunters, young carnivores, have difficulty. Their failure rates in hunts is higher because they don't have experience built into their models to actually be able to have a good predictor of saying this animal will run this way or that way. And that's what makes the prey become better, the predator become better as it ages. Just yesterday, I saw a brilliant video by tennis player Andre Agassi, where he was talking about how he was predicting the tennis serves of Boris Becker. It seems he used to have this very funny tongue movement where he moved his tongue out of his lips, either to the left or to the right, and that signified which way he was going to make the serve. And Boris Becker had no idea that he was doing this. But Andre Agassi watched the videos and he was able to pick up on this and then use that technique to be able to return the serves from Boris Becker. So this is, examples all across animal kingdoms, from cockroaches to tennis players.
All right. So the question from Srihari, was there any difference in time of responses amongst groups and a single fish to a particular stimulus, or any time lag in response to particular stimulus among groups of fishes and a single fish? I think this is the individual versus the...
Can you repeat the question? I didn't get it.
Was there any difference in the timing of response? Let's forget the term time. In the response amongst groups and a single fish, okay, to a particular stimulus.
Okay, so we didn't do experiments where we had multiple fish at the same time, but the latency or the response is more or less within a population because it's a reflex response. The responses occur anywhere from about 200 milliseconds after the flow turns on to about a second and a half, and there is inbuilt variability in the animal itself. Each animal can pick from this distribution. You have a distribution of how quick they respond, and they pick from anywhere from 200 milliseconds to 1.5 seconds. And as I told you, the fish that have very good models inside them, very good predictors, they do better on this side of the distribution, 200 milliseconds shorter latencies, and the fish that are not good predictors will typically have latencies in the longer range. I hope that answered their question.
Yeah, but if not, the person, I encourage Srihari to write back because we'd be happy to clarify if we didn't understand your question correctly. The question now, I think, is more linked to aversive or behavior. If the pattern behavior is, the question being asked is if this pattern behavior is observed in higher organisms, and what kind of emotional response might be seen. Is this also seen in humans, and do fish have an emotional response? Emotion, I suppose one should look at a stimulus which is unpleasant or aversive.
So it's difficult to read out emotions, right? So the only way we can read out is by the response, whether the fish has an attractive or an aversive response. But that doesn't mean that the fish feels anything, which is true for mice as well, or monkeys. When you do experiments on these animals, the only way we can read out is by looking at what they do. But whether what they do comes out of an inner feeling or an inner emotion, the way we feel emotion, that cannot be ascertained at all.
Okay. We now have a question from Lakshmi Perumathwani. My mistake, I inadvertently identified a college as the last name of a person. So thank you, Lakshmi, for correcting that. But she has a question. Lakshmi is also an alumnus of our campus. Post-spinal cord injury, for example, trauma-induced, how does the neuronal response adapt in the cerebellum? And does this have an effect on spinal cord recovery, especially to sensory stimulus?
Right. So there is a lot of adaptation, not only in cerebellum, but even within the spinal cord, posterior to the injury as well as anterior to the injury. And in most cases, depends on the extent of the injury, whether it's a complete transection of the spinal cord or if it's an incomplete injury. Complete transections, it's impossible, or the recovery is poor because we don't have regeneration of axons across the injury site. And so, whatever circuitry is, so if you have a transection, let's say at the thoracic level, everything above that is disconnected from everything below it. And so that's why these people are paralyzed. But if it's an incomplete injury and the injury is small, then it's possible to do physiotherapy and rehabilitation. And what that does is relearn through existing pathways how to control the lower proximity. And that fair amount of recovery is possible there. And that's why, physiotherapy is done to teach the existing neural pathways how to talk to the circuits below the site of the injury.
Excellent. I think we're coming to the last of our questions. This is a question about the Purkinje neurons, which says that what are the other types of stimuli that Purkinje neurons will respond to? And is this restricted to a visual cue?
Wonderful question. No, so Purkinje neurons actually receive inputs from another kind of cells called granule cells. And roughly about half the number of neurons in the human brain are granule cells. They are that many in number, about 50 billion of them. And what they do is they are like switchboard of the brain. They draw inputs from several regions, both sensory as well as motor, from the cortex, from multiple places in the brain, and then they funnel all of this into the Purkinje neuron. So obviously the Purkinje neuron needs to sort out from this smorgasbord of inputs that come in. It responds to vision, sound, it responds to motor efference copies. For example, when the animal is about to make a movement, a copy of that command is sent to the Purkinje neurons. So there are a bunch of different things that Purkinje neurons respond to, and that's why it's still an enigma exactly what they do. All of us are trying to look at it from different facets. If you look for vision-related stimuli, you'll see it. Vision-related responses, you'll see it. If you look for audition-related responses, you'll see it. And so, they are very, very versatile.
Okay, brilliant. Vatsala, there are many notes thanking you for the session. So I think it has resonated with many of our participants. We've covered all the questions, but if there are additional questions, please feel free to write in to Vatsala or even to the Science Setu at instem.res.in email account. We'd be more than happy to have these directed to our presenters. Do you have any closing remarks, Vatsala, at this point that you would like to sort of put on as a take-home message? Or
yeah, so I want to thank all of. I did not imagine that I'll have such a large audience on a Saturday morning, and I'm so overwhelmed by all the questions and all the comments thanking me. It's been my pleasure to talk to this audience. I hope I can do more of such outreach activities. So thank you for coming. Thank you for spending an hour listening to me. And I want to reiterate that, the times may be what they are, but science is what holds hope for all of us. So stay curious, stay safe, be masked, get vaccinated. Bye.
Thank you very, very much, Vatsala. That was a very uplifting and very informative session as well. So with that, we are going to close the meeting and the engagement. Thank you all. See you on the third Saturday now, later this month.
Thank you.
Thanks very much. Thanks, Chantika. We could close.